So welcome to the Genome BC Genomics Forum 2021. I'm Ellie Griffith. I'm the Sector Director for Health at Genome BC. So in this panel session, we're going to build on the discussions this morning that looked at the international landscape around SARS-CoV-2 genomic surveillance. And we're going to bring it back home to BC and discuss some of the history of using genomics here in BC to understand infectious diseases and how this has paved the way for our work on SARS-CoV-2 genomics. So the format for this panel session is we have three guests joining us for this session, and each of them is going to give around a 5-minute background presentation on their favorite genomic surveillance pathogen. And then we will have a discussion and go into a Q&A session. So if you have questions, please enter them into the question section and we will see those. So first, I'm going to briefly introduce our three speakers today. And if you want lots more details on all of their achievements, you can check out the webpage for the event where there's lots more details on their backgrounds. So first, we will have Jennifer Gardy. Jen is the Deputy Director of Surveillance Data and Epidemiology with the Bill and Melinda Gates Foundation. And previously, she worked in BC for many years. Secondly, we'll have Jeffrey Joy. He's a research scientist at the BC Centre for Excellence in HIV/AIDS here in Vancouver. And third, we'll have Mel Crouchton, who's a medical director for the BC CDC Public Health Laboratory. So without further ado, I'm going to pass it over to Jen, who's going to give us a bit of background on genomic surveillance and TB. Take it away.
Thanks so much, Ellie, and it's a pleasure to be here today. So as I'm sure all of you are well aware, the COVID-19 pandemic really has raised the profile of genomic epidemiology. But what a lot of you might not know is that this sort of approach to investigating infectious disease outbreaks and epidemics with whole genome sequencing is actually something that's been around for over a decade. And in British Columbia, we were actually some of the first people worldwide to both develop this technique and then actually routinely apply it to a series of pathogens. And I know Jeff and Mel, who I've known and had the pleasure to work alongside for many, many years, they're going to speak to some of the viral work. But my team at BC CDC and the BC Public Health Laboratory was among the first to apply genomics to an outbreak of bacterial disease, TB in particular. So that's what I'll focus on with my few minutes here. I think it's actually sort of a funny origin story that you can trace really one of the founding moments in bacterial genomic epidemiology to what was essentially a friendly bet between some of the BCPHL lab directors. So the story was back around, I guess it was 2006, 2007, there was an outbreak of tuberculosis that was happening in a British Columbia community. And DNA fingerprinting, which is what we used to use to figure out relatedness between a pair of isolates, showed that this particular outbreak was caused by the same strain of Mycobacterium tuberculosis that had been found in this particular community for years. It was just that it had gone from causing 1 or 2 cases a year to 10, 11, 12 cases. So anybody that studied infectious disease epidemiology knows about the epidemiological triangle, that any disease is going to be a function of the host, of the pathogen, and of the environment. And so at BC CDC and the BC PHL, when the lab team was trying to figure out why there'd been such a sudden increase in cases, some of the team thought it was the pathogen, that something had somehow changed in that strain's genome to make it more transmissible. And other folks thought it was the environment. So around this time, say by about 2008, the Genome Sciences Center was running some of the earliest Illumina sequencers. These were things that were throwing off like 50 base pair reads, pretty teeny. And a group of very, very forward-thinking scientists from BC CDC, BC PHL, the Genome Sciences Center, and SFU said, hey, why don't we sequence a bunch of these TB outbreak isolates and a few of those historical strains, and then maybe we can settle this bet once and for all. And the bet, incidentally, in case you were wondering, was for lunch at the then BC CDC cafeteria. At this point in time, 2008, nobody had ever sequenced multiple bacterial genomes from a single outbreak. What you were starting to see in the literature then was a few papers using next-gen sequencing to sequence maybe a few of the same species that have been collected from different conditions, very much sort of a comparative genomics question, but nobody had done multiple isolates from a single outbreak, and nobody had approached this from an epidemiological frame of mind. So I was recruited to BC CDC fresh out of my postdoc. I'd been working on bacterial pathogen genomics since 2001. When I started my PhD in Fiona Brinkman's lab. And I actually remember my first few weeks on the job at BC CDC in early 2009 as being incredibly interesting. Those of you who can recall great infectious disease events of the past might remember that that corresponded to the earliest days of the pandemic H1N1 influenza outbreak. So I was constantly sitting at my desk making phylogenetic trees with the available data to try and understand what was happening there. And then at the same time, I had this data set of 36 whole Mycobacterium tuberculosis genomes that landed on my desk with essentially the instruction to do something with this. So within a few weeks, our team had partially solved the BET issue. We had shown by looking at the different genomes, comparing the historical strain to the current outbreak strain, that nothing had changed. The change had to be something in the environment that precipitated this dramatic increase in case counts. But we still didn't know what that change was. Now, I had originally pursued a career in infectious disease microbiology because when I was 16, I saw Outbreak with Dustin Hoffman and decided that looked like the most interesting career in the world. It's a terrible way to choose your vocation, like based on a Dustin Hoffman movie, but it did work out for me. I was sitting at the desk with all of this genomic data, and I also had access to very detailed contact tracing data about the connections between the people involved in that outbreak, as well as the shared places that they also spent time at. So I figured, hey, why not try to figure out exactly how the TB infection moved through this social network using a combination of the genomic data and the epidemiological information that I had. Because pathogens, even if it's a really slow growing bug like M. tuberculosis, they mutate. So over the course of, say, my TB infection, if my TB bug develops a mutation and then I transmit my infection to you, your TB bug is going to carry that mutation as well. So based on that principle, I realized that by looking for all of the mutations in each of the genomes, and this was very much a needle in a haystack approach, as over a big outbreak of TB over many years, only just a small handful of letters out of a genome the length of War and Peace will actually change. So it really is needle in a haystack. But with that handful of mutations, by looking for those, by looking for who shared those mutations in common, and looking for overlaps in social contacts and infectious periods, we could map the outbreak exquisitely. So to make a long story short, the outbreak reconstruction showed that an increase in crack cocaine smoking in that community was likely the environmental trigger that was responsible for that surge in cases. We got a New England Journal of Medicine paper describing the outbreak, and really that was one of the kind of founding moments in modern bacterial genomic epidemiology. So I spent the next decade at BC CDC working on the technique further. I had a senior scientist position there. And I was also a faculty member at UBC School of Population and Public Health and held the Canada Research Chair in Public Health Genomics, and was really interested in what can you do with genomics in applied public health to really take that technique further. So at BC CDC, we sequenced more and more TB outbreaks. We eventually ended up sequencing almost all of the TB that occurred in British Columbia over an entire decade. And by looking through all that data, by finding common patterns in how TB spread, we could really shape TB control guidelines and policy in a much more informed and evidence-based way. I worked with modelers to develop software packages to help with this sort of analysis. What could you infer about transmission in an outbreak if you didn't have any epidemiological information, just genomic information alone? I visited public health agencies around the world talking about how to implement routine genomic surveillance, especially for TB in both diagnosis and outbreak reconstruction. I had lots of contact with folks like Sharon Peacock and Bronwyn McInnes, who you heard from this morning, who I still collaborate with to this day. And my team actually did a lot of work on some of the non-technical aspects of genomic epidemiology too. For example, how you best communicate this information out to a clinician or to an epidemiologist, and what are some of the social, what are some of the ethical implications of doing work like this. So when COVID happened and viral genomics played such a vital role in their response, I really think that a lot of the groundwork that we, working on TB, and folks like Mel and Jeff and the whole genomic epidemiology community around the world, that groundwork that we helped to lay was being very apparent in the COVID-19 response, whether it's the technical and analytic aspects of genomics, all the way through to how you use that information for epidemiological action. So I like to think our work on TB played a teeny tiny role, and I really did enjoy the free lunch that I got for helping to settle that way back when.
Thanks, Jen. Let's pass it over to Jeff. Jeff, are you able to share your slides?
How's that?
Good.
Okay, thank you for the introduction, and also thanks very much for the opportunity to speak here today. And thanks, Jen, for that very good introduction to the field. I've been fortunate to work on a lot of different viruses in the last few years, and a lot of that work has been in collaboration with Genome British Columbia, so it means a lot that I get to give this presentation. Just start off by saying, unfortunately, I don't really profit from this, but I do accept Bitcoin and livestock if anyone is interested in sharing some. So let's start off really quickly with a brief introduction to HIV near real-time phylogenetic monitoring, which is something that we've been doing here in British Columbia for the longest, over at the center here. And here what we do is we take advantage of the fact that HIV evolves very rapidly. So we use phylogenetic clustering so that infections that retain a lot of genetic similarity, as Jen just talked about in the bacterial sense, are likely related by recent transmission events. So therefore, clusters of genetically similar infections can represent localized outbreaks. I just want to highlight for everybody that it's important to note that we can't use this technology to point the finger at any one particular individual, because we can never exclude the fact that there could be intervening infections that we don't have a sample from, which is why we consider this at the group level. So we've been doing this, as I said, for a long time. We've been tracking HIV clusters in BC since 2013 and providing the information to public health so that they can actually do something about it to reduce the transmission. And so there's a lot of extractable information that's of public health relevance from these clusters. You can look at the colors to see whether the new infection is drug resistant or not. The size of the circles represent the viral load of the individual. So if you have a high viral load, you know that that's an opportunity for transmission, and it should be suppressed. New cases have an outer circle around them. So if you have a large number of new cases in a cluster in a short period of time, then that's a flag that public health should be notified about it. So yeah, we've been doing this, as I said, since 2013. But in the last 2 years, with the support of Genome BC and also the Public Health Agency of Canada, we've been trying to spread the good word around the country, especially to work with more provinces to help them develop their capacity to do this kind of thing. And then also nationally by combining the data from different provinces where that's possible to do that. And also internationally, we've been trying to assist other countries around the world. So that's the HIV part. Now I'd like to just briefly talk about, again, a little vignette about HCV. This is an older project. I wanted to highlight this one because one thing I really like about this project is that it was a collaboration which worked out really well between the British Columbia Centre for Disease Control, the BC Centre for Excellence, and the United States Centre for Disease Control. And here, what we did is we really looked at using phylogenetic methods, we looked at how HCV, the timing of how it spread in North America. So really briefly, there's a lot of people in North America that are infected with HCV, and they're predominantly my parents' age. So they're all in a particular age category. And it sort of was hypothesized. I mean, in the rest of the world, it's pretty well known how HCV got started. But in North America, we kind of had this idea. The prevailing notion was that it was because the baby boomers were doing a lot of, engaged in a lot of behavioral risk, which didn't really make sense to me that many people were engaging in that kind of a risk behavior and got infected that way. So what we did is we assembled all of the genome sequences of HCV available for North America, and we recovered the timing of the transmission. So what we did is we looked at when did the exponential growth phase of the epidemic really take place, and we showed that it was actually 15 years earlier than would be expected if it was a cause of behavioral risk. So finally, I'll just talk about, to catalyze a discussion later, how we've translated these kinds of methods to the ongoing pandemic that we're all facing now. So using the kind of HCV phylogenetic methods that I just talked about, but slightly more advanced, we've looked at how this virus spread initially got started in North America and Europe. And then also we've looked at how, using the similar kinds of methods, how it got started and spread around here in Canada. We've looked also in a different class of things. We've looked at how the virus is adapting to its new host, us humans, after it has transferred into us, and how it is rapidly accumulating variation, as you would expect something after it's crossed into a new host. And then we've also been developing new methods of clustering to help support contact tracing. And with that, I'll just this is my lab. Thanks to all you guys out there for helping. The CFE lab has been really great at providing the HIV data, sequencing data from drug resistance testing that is foundational to our HIV phylogenetic monitoring efforts, and all of the contributing partners and funding agencies. Thank you very much.
Thanks, Jeff. We'll pass it over to Mel now, who's going to talk a little bit about the genomics response for SARS-CoV-2. Vivian, can you share the slides?
First of all, thanks to Jen and to Jeff. And I'm going to give some perspectives from a public health perspective. If you look back a few years ago, if you spoke to the public health lab and the provincial health services, the focus was only on human genetics and cancer genetics. And then we get this pandemic. And what that has created is some real opportunities built on some of the things that Jen had highlighted. One of the things that has been an incredible challenge in the pandemic is bringing data together. And so this is a public-facing dashboard that is updated daily. And yesterday it showed the total number of cases diagnosed in British Columbia, the currently hospitalized, the currently in critical care, the confirmed deaths, and even the number of vaccines that have been injected into individuals in British Columbia. And it can give you very fine details about who's tested, where they are in the province, and how severe their disease is. And there's other data sets that have been brought together. Bringing these data sets together would normally have taken years. They're updated sometimes in 5 minutes, sometimes daily, and they come from a patchwork of these legacy systems which drive us all crazy. But it does help us respond to the pandemic in a very strategic way. Next slide, please. So genomics was very pivotal in informing some of the policies in early 2020. When we first looked at the cases where we had increases in community transmission, they related to strains that didn't arrive from China. They arrived either from the US or Europe in the case of the dental conference. And They also arrived in the case of the long-term care facility outbreaks. They were related to the long-term care facility outbreaks in Washington state. And then this finding was similar in Quebec as well, where people were thinking about individuals coming in from China. But in fact, even in Quebec, most of the transmissions and introductions occurred from the US and Europe. And then a pivotal moment was detecting a case in an Iranian individual who traveled from Iran, no history of exposure to China. And that person was serendipitously tested because they had a respiratory disease. And that illustrated the transmissibility of the virus and led to some of the issues with in terms of border closures, which are very complicated. Next slide, please. So 2 weeks ago, there was the concern about travelers from India. And below, you can see in yellow, phylogenies related to introductions from travelers with a history of travel from India who had this unique variant. And then you see the spread into the community in blue, which illustrates exactly what's happened with all of the introductions that occur from travel. And the challenge is that we think that the best way is to close our borders, but in fact, it is extremely difficult to close our borders because there's interprovincial travel and we already have many of these variants in the country already. And so we can talk about that during the question period. Next slide. In this slide, you're looking at the lineage fractions for the total samples we've sequenced, and we've sequenced about 23,000 Illumina sequences. And this is from August 2020 to April 21. And you see that there's lineages that expand, lineages that contract. And on the right, you can see an expanding P1 lineage. You can see a slightly expanding lineage that was derived from India, the 617, and a pretty stable state for the one from South Africa, the 135, and then an increase and then a contraction of the 117. But there are real biases in the way this data is presented because WGS is often used for outbreaks and travel versus routine unbiased sequencing, which gives us a sense of what's occurring in the province in real time. Next slide. So one of the challenges in responding to this agent is it's not so much about the phylogenetic trees. If you look at the variants of concern, and the big ones are, of course, the one that was derived from the UK and from Brazil and South Africa, what you see is we focus in on those variants of concern, largely because they're associated with increased transmission. In some cases, increased transmission correlates with increased severity of disease, which can sometimes be difficult to assess. And then the biggest concern is the response to the vaccines or breakthroughs. Yes, thank you. So the other important point, though, is you can see in the colors and the numbers from samples that have been derived from the Philippines, the P2 from Brazil, Nigeria, Europe, India, California, US, New York. And what you essentially do is see these convergent evolutions that are taking place across viral lineages in certain positions. And this is focused largely on the spike. And the important piece for people to understand is to know if it's a VOC or a strain of interest that needs to become a VOC, you need to have the epidemiological linkages and the sufficient statistical power to understand their significance. And that's really one of the challenges. At this point in time, managing the epidemic by pure phylogenies is not as important as really understanding what do these variants do to our immune response and to the efficacy of the vaccines. Next slide. So The real most important point is that pandemic response is really interdisciplinary. And data integration is and has been and continues to be a huge and critical gap. We have these clinical data systems, we have these case data systems, we have these test data systems, we have these administrative data systems, and essentially to understand the impact of COVID and how to respond to it, you need to essentially assemble a COVID cohort and compare that to the general population so you can adjust for underlying comorbidities, medications, outcomes. And so what I've quickly showed you is you have some testing, and that needs to be brought together with the sequencing and the phylogenies and the viral evolution. But ultimately, it is about understanding how those interact. And that's one of Jen's important points. It's that interaction between the genomics and the epidemiology that helps us respond to this virus. And ultimately, it is about bringing together what happens from natural or vaccine-induced immune response and basically transforming this infection, next slide, from a disease that often kills elderly and even some who are very young, and making COVID into a common cold. The probability is we will not be able to eradicate this virus, but what you're trying to do is take the people in red on the left side and basically through natural infection and vaccine-induced immune response, eliminate the severe disease. And that's the end game that we want to achieve. And next slide, please. So the issue for us is pandemic response is interdisciplinary. It's not just the genomics, it's not just the immunology, it's everything coming together. And essentially we are in a five-way race. And in the center is the population who are fed up and tired of health measures that work, by the way, but some people have to go to work and can't abide by them, or some people don't care about them. And some people are holed up in their apartments or houses because they're afraid of the virus. Then you have the virus that is doing what Darwin tells it to do, evolve to survive. And then we have these fabulous vaccines with plenty of controversies that we had a delayed ability to deliver them. So there was a delay in the distribution of the second dose. Then we had all the challenges of giving those vaccines to people and trying to overcome some of the hesitancy that you can see in some parts of the world, including in Canada. And ultimately, the question for us is, while we're doing this in the developed world, is will take a number of years to vaccinate the developing world. And will the virus evolve variants that will be of concern to us? While we think we are in a little bubble of our own, we are in a global village, and genomics helps us understand how to respond in that global village. Thank you.
Thanks, Mel, and thanks everybody. So I think you've done a really great job of showing how using genomics not only has been obviously incredibly important for understanding the SARS-CoV-2 outbreak, but also for past pathogens. And one of the things you mentioned, Mel, was around this kind of need for interdisciplinary pieces to come together. So if you look back to before COVID, from those other pathogens that we've worked on, such as TB and HIV, did you feel the same then about the kind of interdisciplinary needs, or is there something about this pandemic that's really highlighted the need for interdisciplinary work? Mel?
I think the good question. I think the pandemic basically puts our response on steroids, but the same issues were there before. And there was work by Linda Wang and Natalie Prestigiecki, who were heavily involved in trying to understand salmonella transmission, for example, that occurred from food products that were inadequately cooked. And by being able to correlate human salmonella isolates with food products that contain salmonella across the country, we were able to change policy to show that those little food products that look like they're cooked do in fact need to be cooked if you want to prevent transmission. And similarly, we've had international transmissions for a variety of different agents on romaine lettuce. And again, genomics was the tool that you needed to try and answer that kind of a question. So genomics clearly is important. And part of that is the interdisciplinary response. It's bringing together the epidemiology and understanding how this influences policy.
Thank you. And another question I wanted to ask maybe to Jen was if she knew what any kind of the gaps are identified from kind of earlier work, for example, the work with TB, and how has the COVID pandemic been able to help us fill any of those gaps?
Excellent question. Gosh, I could probably speak for an entire morning or afternoon on some of the gaps, but I'll highlight a couple that I think are some of the biggest, and some of these have been alluded to by Mel in particular. I think one of the gaps has always been the challenge in linking genomic data to an epidemiological action. I personally, for the 12 years that I've been doing this, have always said, don't just sequence for sequencing's sake. Sequence because the data, the information you're getting out from that is going to point you to a decision, to an epidemiological action. So in an ideal world, you've got, direct line from genomic data to an action, to a policy. But in reality, it isn't the case. And I think there's a lot of factors that contribute to that. I think one of the biggest contributing factors is that the people that are doing the sequencing, the people that are doing the analyses, and the people that are making the public health decisions are rarely under the same roof. I think BC is a somewhat unique exception, but this has definitely been true elsewhere in the COVID response, where you're seeing people from all over. There are academic university labs pitching into sequencing. There's industry labs, pharma labs that are pitching in. There's government agencies. There are even volunteers, citizen science organizations that are using things like the MinION to generate SARS-CoV-2 data. All of these people are contributing data, but there is no direct line often from their data to a policy decision. But I think there's some other contributing factors to whether genomic data can turn into action. Certainly one of those is whether a sample has enough time and location data, metadata attached to it in order to be able to draw a meaningful epidemiological conclusion. At the very least, we need, a reasonably granular space and time bit of information. If epidemiological action or decision is going to span borders, well, it could be a local health authority, it could be provincial borders, it could be international borders. That's always going to complicate things as well. And I think another aspect of why genomic data doesn't necessarily always contribute to action is, has your agency really committed to making it a part of routine operations? And that certainly varies a lot from jurisdiction to jurisdiction. And in many places, this sort of work is still considered research. And this is a huge gap. Just viewing this as research and having it only done with research funding and not a center's core operating budget, have it being done, labor off the backs of grad students and postdocs, not laboratory technologists, is a big gap. And I think, if you were to do, say, a principal component analysis of the UK's genomic response and why it's been so successful, I think a big driver, one of those principal components, is the fact that Public Health England has routinely been using genomics for everything from flu to enteric diseases to TB for years now. It is an SOP, and it's an SOP built on a strong research community, but one that has really been sort of operationalized. So I'll stop there. I could go on more about data management, but I suspect that will come up later in this session as well.
Yeah, and that's a really interesting point you make about how the information that can be gained from genomics is actually put into action for public health. And Jeff, I wanted to ask you, I think we're all much more familiar now about how the genomic information is used for COVID to inform some of our public health actions. But could you maybe describe a little bit in the kind of HIV or HCV space, how that genomic information is practically used to inform public health measures, especially when you said that you're looking at it at the group level and not at the individual level? You're on mute, please.
Thank you. Yeah, so I mean, at a practical level, by identifying groups that are at high risk of transmission, HIV is kind of a special case because of the need to maintain confidentiality and patient privacy. But by identifying groups that are at high risk of transmission, whether that's an age demographic or a spatial demographic, it allows public health to focus their limited resources where they know the problem is the worst. Yeah, so that's one way.
Great, thank you. I'm just looking at the questions that are coming in through the online, and there's lots of great questions. So I think I'm gonna try and weave some of those in. There's a question here from Evelyn who asks, has the start of vaccination had an effect on the presence of certain variants of SARS-CoV-2 in the community? And is it what you would expect? Perhaps I'll throw that question to Mel.
That's a good question, and it's a complicated question, in part because this pandemic it's a very dynamic process. So every time you're looking at it, you have to look at it in the context of the phases. So Canada made a decision to basically extend the vaccine dosing interval. And it's a controversial decision, and that's been played out in the media everywhere. But what people don't understand is there was good data from the UK that you were protected for about 3 months with about an 80% protection rate. And you could argue what that really means, but it was mostly protection from severe disease and death. And then we had a limited supply. And so what was the analysis that was done was that if you extended it to 4 months, you could vaccinate more people over a longer time period. And the analogy would be if you have 2 grandparents and you have only 2 doses of vaccine, would you give your grandmother 2 doses and leave your grandfather susceptible, or vice versa? And if you did the analysis, if you gave them each a dose, they would likely be protected for the first few months. And then if you look at the literature, what is clear is that for most other vaccines, delaying the second dose improves the immunological response that you get. And I think that that's going to be proven to be the case. The other thing that's important is if you immunize more people faster, you decrease the load of the virus. And recently it's been shown that when you're immunized, even with 1 dose, you reduce the risk of transmission. So it's a culmination of these kinds of things that all come together. And then the question, of course, by the immunologists say, okay, well, you don't make enough neutralizing antibody. But the protective response isn't only neutralizing antibody. And so it's hard to measure that. And it goes back to what I was saying, that the pandemic response is a chunky and clunky interdisciplinary process where you have to get epidemiology together with immunologists, together with public health to try and come up with the right solution. So to answer that, I don't think it was a major driver of variants. The virus is doing what it's supposed to do. The more successful variants will compete and evolve. And the real question will be whether or not there'll be a blowout variant that will knock us all down. But I suspect that that is not likely. And there is the ability to change the vaccines if that should be the case.
Thank you. There's a question that's very related to that about, is the genomic data in BC being used to actually improve vaccine design? So in other words, are we changing or able to change the vaccinations as new variants emerge?
So this, I think the last time I looked on GSAID, there were more than 1.2 million published sequences. So globally, even in the most remote areas, they're doing sequences to look at the various strains. And all of that material is being looked at to determine whether or not there are significant changes that can justify modifying the vaccines. And in fact, both Moderna and all of the other manufacturers have already produced strains that have slightly different spike mutations to try and anticipate the potential for a variant to impact the vaccine immune response.
Thank you. There's a really great question here. I think, Jen, you can probably address it best about H1N1 from Evelyn again. And why do you think that the H1N1 pandemic didn't reach the same extent as COVID has? Like, what is different about COVID?
Yeah, quite a few things. I think, one of them is that we've always expected flu pandemics. When you look at any pandemic preparedness and response exercise, an influenza virus, a novel reassortant influenza virus is always one of the major candidates there. And so it's an area that our response has already been primed for. So for example, the vaccine production infrastructure for flu vaccines, it's set. It's very, very well established. So the second this novel virus was picked up, that could kick into high gear and we were able to get vaccine out quicker. That pandemic, I think the first detection of H1N1 was around late April 2009, the first detection and public announcement anyway. There was some, had likely been circulating for a few months before in Mexico. But first public detection was end of April, and by October, we were seeing enough vaccine rolling out into communities that we could sort of blunt things. And then you're just dealing with a different virus as well. There's different epidemic dynamics, there's slightly different modes of transmission, and it is just in general a lot easier to deal with a known entity. This coronavirus, again, pandemic preparedness and response exercises have also often included novel coronaviruses as new disease agents, but people really did have a lot of their eggs in the flu basket. So I think we were a lot better prepared there. But yeah, it comes down to preparedness, just differences in how the viruses transmit, difference in their R naught, and yeah, just how novel they are, pre-existing immunity as well. If you'd been exposed to a seasonal flu in years gone by, there was likely going to be some cross-protective antibodies. So quite a few factors combined to make H1N1 less of a concern than COVID.
I just want to add a little point to what Jen said. So with H1N1, the older population was immune imprinted and exposed to many of the immunodominant epitopes that were important to control. And so the elderly were actually not very affected. It was more the younger people who'd never seen this virus before. And so you had a totally different dynamic. Whereas COVID, if you look globally, everywhere in the world, there's this really clear correlation between age, severe disease, death, and that is very interesting. And in terms of one of the things that's also very interesting is there was a number of publications showing correlations with these endemic coronaviruses, which share some of the sequence homology. And if you've had a recent endemic coronavirus, you're less likely to have severe disease. So there's something that is related to those endemic coronaviruses that can reduce the impact of COVID, but COVID has figured out how to really harm the globe. And it's really a challenge to understand the immunology in its fullest context.
And Mel's example of what happened with H1N1 and realizing that elderly people, folks that had been around in the late 1910s, early 1920s, had this sort of cross protection, was actually a really nice example of genomics indicating that. It was very early phylogenetic analysis that revealed that the hemagglutinin and neuraminidase, these immunodominant epitopes that Mel mentioned, they were most similar to the H1N1, the pandemic 1918 strain. They'd sort of jumped into swine and hung out and evolved really slowly there. And so genomics provided a really nice explanation for this biological observation.
Right. So a question from Federica. So there's a notion that in BC we are no longer sequencing all of the genome. So how are we tracking the variants for direct input into public health measures?
We're doing a combination of SNP analyses in some locations, but we've done some calculations, and our sequencing capacity is pretty high. It's about 1,900 a week. And we've done some calculations to sample in an unbiased way from all regions of the province. And with that sampling strategy, we believe we'll be able to detect as little as 1 in 200 or 1 in 500 of some variants that might occur. And again, what our predominant interest at this point is not so much the phylogenies, but understanding if you have if you have certain variants, how does this impact the vaccine response? And we are actually integrating the data in British Columbia in real time. It's very, very difficult to do, but basically who's tested, what their result is, what is their genomic profile, how is it impacted by the presence of a variant, and how is the vaccine, what's the performance of that vaccine over time?
Right. So somebody is wanting to know, Evelyn, is the whole genome sequencing data made available in public repositories?
We try to do that, and one of the challenges we have is resources. So even in compiling this data, the systems we use are painful, to say the least. And bringing them together and then putting them into a public repository, we try to do the best. And federally, they've just initiated a portal where we can try and share the data. But even there, I think Jen highlighted a very important point. Genome England made some strategic investments over a decade to create infrastructure that then got used for the pandemic. And we're trying to respond to the pandemic, create an airplane while we're flying it. And it's very, very difficult to do that. And that's one of the challenges in our country, which is 13 countries in one, 3 territories and 10 provinces. It is hard to get some of this stuff aligned.
That goes back to the gap issue that we were talking about earlier, and I think this is certainly one of the biggest ones is data management and data sharing. When you look at public health genomic epidemiology, I think data management, just how does this data get analyzed? How does it get shared? It has always been I'm just going to use the word disaster pretty much every level of the system. Mel's laughing out of recognition. Within an individual agency, for example, you've got data management being done sometimes on commercial software platforms that other agencies can't afford. Sometimes it's on open source platforms. In many of those commercial platforms, there might not even be a way to store genomic data or even abstracted genomic data. Like this isolate belongs to outbreak X. There's if you go into most laboratory information systems, there's not even going to be a field saying, hey, enter the pangolin lineage for this SARS-CoV-2 sample. So everything just kind of ends up either in Excel spreadsheets full of metadata in generally non-interoperable formats. There are efforts like Public Health Alliance for Genomic Epidemiology that are trying to get around this. And BC and BC CDC and BC PHL staff in particular have done a phenomenal job of trying to contribute to better interoperability and standardization of metadata. And then when it comes to public release, it's all over the map. I mean, some people are releasing raw FASTQ files to something like NCBI's SRA. I personally, with all the TB work that we did, every FASTQ file went into SRA with some pretty decent metadata attached to it. Some people would only publish assemblies, consensus sequences, and so you have no idea what the upstream bioinformatics leading into that was. And then sometimes you've got specialized repositories like GISAID for flu and for SARS-CoV-2, but for other diseases, there is no central global warehouse. So you've got this weird system that's sort of built on fundamentally broken and fragmented software blocks. And I think it is one of the things that really is standing in the way of making genomic epidemiology something that is routinely used within a public health agency environment. But it is something that I think this particular pandemic has been particularly instructive for. I think the COG UK consortium has done a really fantastic job, wherein they've got a single centralized bioinformatics platform, a cloud-based platform that all the raw data is flowing into, that is running the same analyses on all of the samples, regardless which lab they came in from, and regularly pushing out to the databases that data needs to be in, including just making it available through a web portal and creating a number of visual front ends to that too, because there's a lot of information that you can glean from pathogen genomics and phylogenomics that can be used by anybody. It doesn't have to be, you know, a Jeff Joy with an amazing series of degrees and track records in evolutionary biology. But when you present the information visually in a simple, intuitive, interpretable way, a lot of people can draw useful conclusions from this data. So I think we will get better. I think this has really been a catalytic event in improving the way we approach data management, but it has certainly been one of the biggest problems to date.
Yeah, we keep.
I just want to add one little thing. So, you know, one thing that the public is not totally aware about is we know that health measures hurt, and some of the decisions around, do you close a gym, or do you close a restaurant, or do you close singing or whatever, and those things are informed by the epidemiology and less so by necessarily the specific phylogenetic tree. And the challenge is, like, Whistler was not closed because of the mountain. Whistler was closed because of the people who worked there, lived in congregate settings and would go and drink in bars and spread virus. And people hate that. But it's the only way you can maintain enough reduction in transmission until the vaccines get into people. And that's an important challenge for us as a society, to try to do as minimum amount of harm to the society while you're trying to respond to the pandemic.
So talking about the kind of vaccination strategies and the difference they're making, and Mel, in your presentation, you referred to the fact that the goal might be that this becomes like the common cold or even like flu, I guess. Does that mean moving forward, we might expect to have ongoing annual vaccinations for COVID?
The reality is nobody knows. We know that if you immunize individuals, and in Israel, they have already shown that you get a decline of antibody after about 6 months. And so some of the early findings are going to come from the UK and Israel and even the United States, where they've vaccinated so many individuals. And again, we're going to need to look at what the efficacy of the vaccine is. So who gets reinfected, who gets severe disease, and then make those immunological correlates to determine whether or not a booster makes sense or not. And that's the challenge for us at a global level. And then at the same time, figuring out how to support economies like India and some of South America, who are really having big trouble with this infection.
Yeah, talking of India, and Jen, I know you have a great kind of global perspective on infectious diseases. There's a question here from Lily asking, so last year when many countries were really in the firing line for COVID, India was doing pretty great overall. Do you have any insights into what factors have contributed to the current more challenging situation in India right now?
I think really it just boils down to what is so often the case in pandemic response, which is just a series of Swiss cheese holes lining up in the worst possible way. And almost every jurisdiction around the world, with very few exceptions, has been the Swiss cheese at some point. I think a lot of it really does stem from leadership choices. If you look at the first wave in India, there was a very, very strong and comprehensive lockdown, which worked. It did what it needed to do and it flattened the curve. But you didn't see that same response recently. As you started to see cases uptick, the focus was more on reopening and not disrupting things. So leadership issues, supply chain issues have been huge as well, especially when it comes to things like PPE. India is really interesting because it's got its own very robust domestic vaccine production infrastructure, and it has in fact actually been supplying vaccines to the UK. And I'm not familiar with the exact nature of what proportion of domestic infrastructure, domestic vaccine production was being kept in India, but perhaps it was too little. And now we're in a situation where other countries around the world are committing everything across the commodity spectrum, from PPE to the reagents, the consumables, the lab equipment that you need for testing, through to doses of vaccines as well. So it really was a perfect storm, and a perfect storm in the perfect breeding grounds for COVID transmission, which is very densely populated and oftentimes multi-generational living situations. A lot of people have wondered, why hasn't Africa been hit to the same extent? And it's actually got a similar pattern to India, where the first wave was not as bad as we expected, but you're currently seeing a bit of a surge there. It's certainly not a surge to the extent that we're seeing in India, but again, it's a lot of the same issues. I think with Africa, you've got a bit of a buffer in that it tends to be a much younger population as well, so there's a demographic effect at play. But yeah, in many countries around the world, you've had a successful first wave, and then the second wave or third wave in some cases, some fourth wave in other cases, has really reared back in a way that people didn't anticipate. And I personally am cautiously optimistic. I also think cautiously optimistic is like my phrase of the pandemic. I'm cautiously optimistic with the way vaccination is going, and just within the last couple of weeks, even decision to make more vaccine available to countries that are experiencing surges like India, like parts of Africa, really accelerating the global effort. Maybe this will be the last phase of surges. As Mel pointed out, you can never predict anything with a pandemic. We have no idea what's going to happen. But my sort of educated infectious disease epidemiologist guess would be that we are coming to a point where future surges will be sort of law of diminishing returns.
Yeah, can I just add, so I completely agree with what Jen has said, and then I think what has gone on in Brazil is a very interesting example. So in Manaus in Brazil, where they had estimated that 60–70% of the population had been exposed to natural infection, they then had a very, very severe next wave. And it's precisely for the reasons that Jen highlighted. And it's illustrative of the value of the vaccine, because they haven't been able to vaccinate effectively there, it basically perpetuates all of the challenges that Jen highlighted. And another thing in India, we became terrified with this variant from India, the 617. But if we look at transmissions, people who are coming into Canada, many of them have the UK variant. So it's not clear to us that, in fact, it's that variant from India that is causing some of this devastation. They may well be affected by the UK variant extensively. And we know that that is more transmissible. And it's all of the points that Jen brought together, which is an illustration again that pandemic response is interdisciplinary, and the politics of pandemic response is very, very crucial to really bring those pieces together. And it's not trivial in trying to come up with the right solution, and people make mistakes, but you have to learn from those mistakes, and it's really important to look for the signals.
Thanks. And obviously there's a lot of discussion in everybody's lives right now about vaccinations. And I wanted to ask Jeff, as we're talking a lot about vaccination, how quickly we've managed really to roll out new vaccines for SARS-CoV-2. But we don't hear about vaccination in the HIV space. And I was wondering if you had any insights as to why that is, and is it anything to do with the genome of HIV as to why we don't have vaccinations in the same way that we've rolled them out currently?
Yeah. So, I mean, one thing is everybody's talking about how, you know, they haven't got their vaccine for SARS-CoV-2 yet. I think another way to look at that is that you already have the opportunity to get a vaccine, and we have to be a little bit patient with that, because it's actually astonishing that we have not one, but a whole bunch of different vaccines that are effective and useful. So yeah, I think that's one thing that could be said more, is that, yeah, yes, it'd be nice to be vaccinated, but another way to look at that is most other things, it takes a lot longer to develop a vaccine. And secondly, you know, things like HIV and HCV, we just don't have a foreseeable pathway to having that, even after years and years and years and years and years of those pandemics being a thing. To get to your question about why, you know, those two viruses, HIV and HCV, they are some of the fastest evolving things that we know. So they evolve extremely rapidly. They recombine. Well, HIV is also programmed to recombine. So that just makes it very difficult to develop an effective vaccine. You know, it's possible that a population-specific vaccine might be something that could work for HIV one day, but the real difference there is the two viruses, HIV and HCV, move so much more quickly that by the time any kind of vaccine is developed, the virus has already moved too far away from it.
So can we consider ourselves fortunate for COVID that it doesn't mutate and evolve as rapidly as HIV and HCV?
Well, it doesn't evolve as rapidly, but it does evolve, and it is accumulating variation. If you plot variation that's accumulating, it's accumulating in a way that is useful for us to do things like molecular epidemiology. But yeah, we can consider ourselves fortunate that it is not mutating as rapidly as HIV or HCV.
Mm-hmm. So coming back to the online questions, one that's getting quite a few votes here from Li Shuang. Most Canada SARS-CoV-2 data shared in GISAID only reported collection dates at month, but not day level. Any regulation that limit the sharing of exact day, or are there any limits as to why we can't share the exact day? Because we need to have the collection days to use the data for transmission inference. Is anybody, Mel, are you able to?
Initially that was a battle where we were told that for privacy purposes, initially we were not to include the date of collection, just because we had just a very few number of individuals and they felt that would identify the individual. I think we've grown beyond that. So everything that we do release now has the date of collection, and as it's been pointed out, the date of collection is so crucial to determine what's going on. I think the challenge for us now is really being able to look at the data from a national perspective, to understand and to power the epidemiology so that you can actually look at the impact of the variants on the vaccine effectiveness. It's less about the specific transmissions, even though I think that people are fixated on that. But it's really about understanding, okay, if you're vaccinated, how well are you protected? What are the features of the virus that evade? and then really trying to understand in a very sophisticated way, as Jen really highlighted, it's that triangle of the environment, the virus, and the host.
And I think, you know, it's sexy to focus on transmission inference. It's really cool. It's that, you know, Dustin Hoffman and outbreak sort of thing. But the breadth of epidemiological questions that you can address with genomics is so much broader than transmission alone. And it also varies at different stages of an outbreak. And the granularity of the metadata that would be required at different stages varies as well. Unfortunately, it sort of varies in the inverse. So the most detailed data is the most useful at the very beginning of the pandemic and towards the end when you're mopping up outbreak flare-ups. And that's when there is the biggest risk of being able to link a specific viral sequence in a database to a specific individual. You know, for example, if you look at, there's absolutely groundbreaking work that was coming out of Trevor Bedford's lab all throughout the pandemic, but particularly in the early days when the first detection in Washington state happened. And then the second detection came along a couple weeks later. The Bedford lab was able to access those two samples, did whole genome sequencing, and realized that the genetic distance between those samples indicated that there was this sort of period of cryptic transition or transmission within the community. And the issue was that like, we know who patient one was. Their information was front page news in Seattle newspapers for weeks. And there was a very good idea about who patient two was as well. So you could very clearly link a specific sequence to a specific individual. And that will happen again too. I mean, if you look at one of the first really big rollouts of genomic epidemiology in real time in outbreak management, it was when Ebola hit West Africa, 2014, 2015 was sort of the bulk of the outbreak. And in the waning days when it was really all about determining why an outbreak had flared up in a particular region, many incubation periods after the last case in that area, again, you got to this point where you did have the ability to identify very specific individuals. Unfortunately, for most jurisdictions right now, where we're sitting in terms of the burden of COVID-19 disease means that transmission inference is impossible. There's just so much community transmission happening. It really is limited to just say a workplace outbreak investigation. But we are going to reach a time, it's going to be different depending on which jurisdiction you're in, but where the cases are few and far between, that information does become identifiable again.
A quick question to throw to Mel before we have to start thinking about wrapping up this session. This morning we heard about the sequencing rates for especially the US and the UK. Are you able to kind of share with us what our current sequencing rates are here in BC? You're on mute.
The sequencing rate, so we're doing about 1,900 genomes every single week. And we think we can even increase it. The sequencing isn't the obstacle, it's everything else. The data integration, the epidemiology, the linkages to the points that Jen highlighted, the points that Jeff highlighted, that's the challenge for us.
Mm-hmm. Great, thank you. So just before we wrap up, I wanted to just kind of look to the future a little bit. We've talked about the past and some of the past work we've done here in BC and around the world, and obviously focused a lot on talking about COVID. But I'm going to ask you each, and you can answer it quite briefly, but in your opinion, what infectious diseases that we already know about do you think have a real unfulfilled kind of need for genomics, like where the potential of genomics has not yet been realized in how we're able to tackle those infectious diseases? And maybe I will start by going to Jen, because I think she feels quite passionately about this.
Yes, I do. So it was about 2.5 years ago now that I left BC to join the Gates Foundation in a leadership role on the malaria team. And really a big part of my team's strategy and investment is taking what we've learned, both the good and the bad, from the past decade plus of bacterial and viral genomic epidemiology scale up, and really porting those lessons to the malaria space, both on the parasite side and the vector side. I actually work very closely with Bronwyn McInnes, who you all heard from this morning on a lot of malaria genomics work. And what we're seeing is that there is actually a tremendous amount of sequencing capacity and capability in places like Africa. And the Africa Center for Disease Control, which is about 4 years old now, is really committed to leapfrogging over the genomic training wheel years and really implementing pathogen genomics at scale for diseases that matter to the member states of the African Union, including diseases like malaria. So personally, I'm most excited about how genomic epidemiology is really going to new places around the globe, how it's going to be generating information that feeds directly into things like a national malaria control program's planning and decision making, and how international scientists are really coming to global prominence in this area and are showing that this is a technology that isn't just for places like Canada and the US and the UK.
Great, thank you. Jeff, do you have any kind of insights or favorite?
Yeah, I'm also quite passionate about this. So yes, I guess what I would like to say, I'm not on mute, am I? No. Is that, as climate change expands the range of vectors and the pathogens that they carry, and as human development continues to encroach on wildlife populations more and more, we're going to see spillover events like this one that we're faced with now more frequently in the future. I think that it's really highlighted the value of genomic surveillance architectures at a broad scale as well as at a fine scale. And in terms of what vectors or what pathogens do I think are going to be important, I think that increasingly we're going to come into contact with bat and rodent populations more and more, and that the viruses that circulate in those those animals are the ones that are likely to spill over. So possibly things like maybe Lassa fever could become more of a thing. Nipah virus could become more of a thing. And all of this points to the need for better surveillance architecture at global, national and regional scales.
Thank you. Mel, final word from you on
So that was perfect. So after Jen solves malaria, and I completely agree actually with Jeff about climate change, because if we think the pandemic it requires interdisciplinary behavior, climate change requires a much more sophisticated activity. And I do agree with Jeff that the future is about One Health, and essentially being able to understand how climate change and globalization really will change our society in terms of human, animal, food, and environmental interactions. And then Jeff is correct. I think we need to have this broad global response capacity. Canada needs to up its act to have a more integrated structure, and we've had discussions about that. And then locally, you need to have some of this capacity. And I'm sure data sharing will be on our minds for decades to come, but eventually we will get there.
Thank you. So our time is up. I just want to say a huge thank you to all three of you. It's been a really fascinating discussion, hoping that perhaps at some point in the future we can do this in person rather than on Zoom. That would be really nice. Yeah, thank you for this, and hope you're able to join some of our other sessions as well, not just as a panelist, but as an audience member. And I'm going to say goodbye now and hand it over to Pascal.
Pascal, you're on mute.
I will start all over again because that was not part of my instructions. But anyways, thank you so much for the speakers, panelists, and moderators today. And it has been a fascinating first day of our Genomics Forum. And also give an enthusiastic shout out to the Genome BC staff who in the background have made these sessions possible and will continue to do so over the next 2 days. And thank you to our audience as well, to you for participating in this first day of the 2021 Genomics Forum. Following these fascinating talks today with a focus on genomic sciences in the fight against COVID-19, we will shed the light on vaccines and vaccinations tomorrow. And on day 3, we will discuss the role of media, social media, and science communication during a pandemic. So for tomorrow morning, we will start at 8:30 PDT, Pacific Daylight Time, and we will open the day with Dr. Anthony Fauci. the director of the US National Institute of Allergy and Infectious Diseases and chief medical advisor to the US President, who will be in conversation with Dr. Peggy Johnston, vice chair of the Genome BC Board of Directors. To join this session, please go on the Genome BC event hub and click View Session. I look forward to seeing you tomorrow for day 2 of the 2021 Genomics Forum, and enjoy the rest of your day. Thank you. Bye-bye.